Nothing closer to my heart is to look at the scientific toolbox and the fundamentals, the technology and the innovation in cell and gene. That there are still lots of bottlenecks to be overcome when it comes to driving efficiency and cost of goods. And I thought that there's no better way to do this than spending a bit of time back on the bench. And I've had the privilege to found a great collaboration with the National University of Singapore. I have an amazing team of scientists that I'm working with to hopefully develop some consequential modalities and toolbox when it comes to gene transfer. And hopefully these will result in some significant step changes when it comes to manufacturing efficiencies, precision and safety, and ultimately cost of goods as well.
Welcome back to part 2 with Lucas Chan, who's a fellow of the Royal Society of Biology, an advisory member for Singapore Ministry of Health, and a viral vector manufacturing pioneer. If part 1 inspired you about the cell and gene therapy revolution, you'll love this technical deep dive. We'll explore strategies for successful cell and gene therapy manufacturing and ways to make these therapies more accessible and affordable. Plus, Lucas shares his entrepreneurial insights and vision for back to basics in innovation in his consulting journey. Ready for game-changing solutions? Let's dive in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Now, we need to look at yet another aspect because, yes, it's complex, not as mature as biologics, for instance, but something we often hear are the costs, and that's a big challenge. Can you tell us what innovations you're most excited about and how they potentially could help us drive down the cost of cell and gene therapy and thus enable a wider spread access?
Again, I will fall back into my cushion line of everything, everywhere, all at once. Until you solve every aspect of cell and gene, nothing is really solved. And I think that is actually also applied when it comes to cost of goods. When we talk about cost of goods, obviously, particularly in the emerging markets of APAC, where the demographics are very different, the affordability criteria is also very different compared with, I guess, more advanced economies in Europe and US. And this is where it is acutely felt when it comes to discussing market penetration and market access. But if I could deal with fundamentals of what drive costs in cell and gene, invariably is the complexity of manufacture. Particularly to date, what I believe we're going through version 2, I guess, in the cell and gene life cycle, if I may put it that way. We've already seen market authorization of first cell and gene products receiving approval. We've seen transformational effects in efficacy in some of this cell and gene therapy in the treatment of rare diseases, and now not so common rare diseases, but single gene disorders, to hemato-oncology and even towards solid tumors. So now I think what we're dealing with is finding efficiency. These are very complex manufacturing process. They have many multiple steps. There are a lot of raw materials. It's a very complicated manufacturing process to navigate. So in a way to find efficiency is to break down the process, looking at the fundamentals. One example in viral vectors at the moment, they're commonly produced through transient transfection. again, is a very difficult part of the manufacturing process to control. We're now seeing examples of stable producer cell lines that are being rolled out. And this is big modality, a big technological advancement that we keep looking forward to seeing better and more producible cell lines that have become more widely available. So for me, the cost of goods is ultimately driven in cell and gene by the question regarding efficiency. And efficiency is finding efficiency through the current best practices. Again, we're still borrowing technologies from traditional biologics manufacturing, but I'm now seeing more customized cell and gene technologies being rolled out, both upstream and downstream and at a molecular cellular level. So these are really encouraging to see. And I think this is where the fundamental question about finding efficiencies is going to be the big question I think developers are constantly challenging and thinking about. And whether we can develop new gene transfer modalities that is easier to manufacture, like, for example, in cell line development, or do we think about fundamentally how we genetically modify the cells in a different ways? We now have very successful examples using non-viral modalities, perhaps in larger inserts going into cells. So all of these will contribute to what's ultimately driving the cost of goods into manufacturing cell and gene therapies with greater level of efficiencies and without obviously compromising good manufacturing practice. And ultimately that means safety.
I'd like to change gear a bit here, Lucas, and talk also about your leadership experience, because you mentioned that there are a lot of moving parts to manage. I mean, the technology is very complex and you have a lot of raw materials and a lot more is going on. You have a lot of different people you have to manage. And previously you were working as a CSO and in the CDMO. So what leadership advice can you give our listeners? Let's just give the top one leadership advice that helps you succeed in this complicated role.
If I boil it down to some of the key priorities in terms of my communication, in my shared learn experience with my colleagues that I've had the privilege to guide and to work with, particularly in cell and gene, I think I work with a lot of colleagues that came from traditional biologics manufacture, from advanced research institutes. It's really to help them remember and think about what is it that they are dealing with and why it is that they're dealing with it. You don't have to be involved in cell and gene. You can do other things in life. Why is it that you want to come into this tremendously frustrating, but at the same time, very rewarding field to be in? And to explain and to guide colleagues in getting them to understand. So how is what they're doing relevant to this ultimate mission, which is, in my personal view, there's no greater reward is to see some of these transformative pipeline development entering the clinic. It is to try to get them to understand that little part that they play is fundamentally very consequential in what is going to come out the other end.
And what have you seen as you were starting the company and then eventually developing it? Because many of our listeners dream of translating their scientific expertise into entrepreneurship, whether it's by spinning off a technology, whether it's by starting a completely new startup. What advice would you give to science considering founding a biotech company, especially in new markets like you have done?
I think if I could boil it down again to a couple of learned experience I have had the privilege to gain along the way, is to try and speak to others who perhaps has done a little bit more than you, who's gone through the process and who's found successes, but also lessons learned that hasn't done so well. Don't try and reinvent the wheel if there are already others who's tried to do it, try to gain and learn from someone who's perhaps had a slightly longer journey than yourself. And I think, as I mentioned before, cell and gene is a very interesting field to be in, and APAC is an emerging market and have different affordability criteria. Now, I don't have answers to all of these questions, and I think it's really important to remember that cell and gene is everything everywhere all at once. Maybe not one person can do. It really is a village effort, it's a community effort, it's a stakeholder coming together to work together. It's a really enjoyable journey to work with other subject matter experts who's within the area.
Before we continue, biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, "David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs." Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode.
Of interest.
And speaking of journey, Lucas, you are now on a journey. You just recently started a new chapter. Tell us a bit what you are doing right now and why this new step.
Yes, so I thought there were a couple of things that I'd like to focus on in my next chapter, and nothing closer to my heart is to look at the scientific toolbox and the fundamentals, the technology and the innovation in cell and gene. That there are still lots of bottlenecks to be overcome when it comes to driving efficiency and cost of goods. And I thought that there's no better way to do this than spending a bit of time back on the bench. And I've had the privilege to found a great collaboration with the National University of Singapore. I have an amazing team of scientists that I'm working with to hopefully develop some consequential modalities and toolbox when it comes to gene transfer. And hopefully these will result in some significant step changes when it comes to manufacturing efficiencies, precision and safety, and ultimately cost of goods as well. The other chapter that I'm also embarking on is my consultancy journey, which I felt that, as I mentioned before, cell and gene is really a village effort, and it's really important we interact with each other, learn from each other and support each other. And I like to dedicate some of my time for developers and other stakeholders within the community. Perhaps might have a translational issue, they may have a scaling issue, or GMP and a compliance and regulatory issue, or even commercialization risk of certain fundamental technologies. I'd like to impart what I've learned in my personal journey to see if I can make an impact on their journey.
Yeah, that's fantastic. Thank you for this. And also, pouring your expertise and knowledge into the wider cell and gene therapy community. I think we need a lot of experts. We need a lot of different people, and we need to solve these big problems together as a community. So before we wrap up, Lucas, what burning question haven't I asked that you are eager to share with our biotech community?
I think you've burned through everything that I can offer. So 2 areas we haven't covered, which is process analytics technology and also safety when it comes to biosafety, replication competent vectors. So perhaps I could mention just one of them, I think on RCL, which I think quite a lot of listeners will be interested in, which is how is it that lentiviral vectors are safe when it comes to a non-pathogenic and wild type point of view. So I think it comes down to the fundamental design of the virus. It is no longer an intact virus. The DNA has been split up into multiple cassettes, into different expression helper plasmids that we bring back together in the cell line to recreate a virus-like entity that only does one thing, which is deliver the therapeutic gene of interest into cells. It doesn't replicate. Fundamentally, it is unable to replicate because we have removed the viral genes that are critical in the process of replication. There are other technologies that's been designed in the pioneers of lentiviral systems in the early 90s, which is the introduction of a self-inactivation system. Essentially, what that does is that once a viral vector goes into a cell, it's a disable switch that it then disables itself and unable to express its genes of interest. So it's a very clever mechanism that is built into the vectors without affecting the therapeutic transgene of interest being expressed in your target cell of interest. So I think that fundamentally makes the system extremely safe. And on some of the more recent reports, thinking how many patients has now received a gene-modified cellular therapy, particularly the ones that use viral vectors or lentiviruses, it's gone into the thousands, if not the tens of thousands. And as far as my knowledge is concerned, there hasn't been any reported case of detection or the occurrence of any pathogenic or reversion into a replication competent form. So it's a very safe vehicle to use. We're still improving. There's no compromise in safety, how we develop safer, more precise vectors for the future.
As we are wrapping up, Lucas, what is your 20-second takeaway, the number one takeaway you want our listeners to walk away with?
You got me there. I find it's really important to keep asking questions, to keep challenging, I guess, with the younger neuroscientists that are coming into the field, keep challenging the norm, asking why we're doing certain things this way, why are vectors made this way? There are so many disadvantages and so many bottlenecks. What can we do better? So we need to continue to innovate, and I'm continuing this journey.
Fantastic. Thank you so much, Lucas, for sharing your knowledge, your expertise, your passion. It's been a wonderful conversation. Where can people get a hold of you?
So you can reach out to me through David, my wonderful host, but also on my LinkedIn web link. That's the best way to reach out and have a conversation.
Fantastic. Smart Biotech Scientist, please use the link down in the description. Please reach out to Lucas. And Lucas, thank you once again for being on the show today.
It's a real pleasure. Thank you, David, for being a fantastic host and keeping me in check in my time.
These were game-changing insights from Lucas Chan on viral vector manufacturing and entrepreneurship. From scale-up challenges to cost reduction strategies and leadership lessons, we have covered breakthrough approaches that could transform patient access. Remember, viral vector development doesn't have to be overwhelming. Do you need expert guidance to accelerate your process development and cut months off your timeline? Schedule your free consultation at BrühlmannConsulting.com, where we help you turn complex bioprocessing challenges into clear, actionable solutions. Thank you very much for tuning in today. Please leave a review on the platform you found us on. Thank you so much for doing that, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.